Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami, microbiologist. Today in this video lecture we are going to see another biochemical test, that is acetate utilization test. In the previous video lecture, if you guys remember, we talked different biochemical tests, and if you missed, so you can go back to my channel and watch biochemical test. Okay, so we already talked 16 biochemical test. Now it's time to talk the 17th biochemical test, that is acetate utilization test. Now what is acetate utilization test? Again, it is the test that is used to identify the bacteria, mostly aerobic bacteria, that can utilize or that can use water, that can use acetate, that is the molecule or ingredient that is present in the media, mostly in the sodium acetate medium. So if we want to isolate those type of the bacteria that are using acetate ingredient, so then we will perform this type of the test. Next point says that this test is mostly similar to another test which we call that acetamide utilization test. Inshallah, we will also explain this test in some other video lecture in detail. Okay, and due to the similarity of this test, we can isolate and differentiate the bacteria on the basis of their metabolic activity, or we can say by utilization of acetamide we can differentiate the bacteria. Moreover, this test is also used to differentiate Shigella as well as the E. coli bacteria. We better know that both of these two bacteria, Shigella as well as E. coli, belongs to the family of Enterobacteriaceae, and both are the rod-shaped gram-negative bacteria, lactose fermenting bacteria. So if we want to differentiate these two bacteria from each other, so then we have to perform acetate utilization test. Okay, now the next is that one more thing that I must tell you, that Shigella is the acetate negative and E. coli is positive. It utilizes the acetate while Shigella doesn't. Next point says that this acetate utilization test is also similar to the citrate test, which I already explained. You can go back to my channel and watch the citrate test, or there will be a link in the description box which is the biochemical test. You can also click on the link and watch this video lecture. Okay, so it says, this point says that acetate utilization is also similar to the citrate test. Now why? Because in this case we can also see the bacteria that utilizing the citrate, but in this case we are seeing the bacteria that is utilizing the acetate, and these two things, citrate and acetate, are actually using as a source of carbon. Okay, next point says that in this test we mostly use the media which we call it sodium acetate media. Now then we have the objective of the test. So this test, the main objective of the test is that to differentiate between gram-negative rods and also oxidase negative non-motile aerogenic, that is either E. coli or Shigella. Okay. Moreover, we can also use this test for lactose non-fermenting bacteria like gram-negative bacteria from fermenting bacteria. So these are the objective. Let's see the uses. So this test is mostly used to check the ability of the bacteria, that which type of the bacteria can be easily utilize the acetate that is acting as a source of carbon. The first point. Second point says that this test is acting as a qualitative test. Now why? Because it is used to differentiate gram-negative bacteria into fermenting and oxidative group of the bacteria. That is why we can say it is the qualitative test. Next point says that in this test we mostly isolate the E. coli. Okay, that's why this test is important. Now let's see the composition. So as we talked that in this test we mostly use sodium acetate media. So the composition of this media is that it contains sodium acetate, that is 2 gram, magnesium sulfate, that is 0.1 gram, sodium chloride, that is the 5 gram, ammonium dihydrogen sulfate, that is 1 gram, agar is 20 gram, uh, bromothymol blue indicator is 0.8 gram, and distilled water is 1000 ml. Now all these ingredients we will mix it in 1000 ml, and then we will get the sodium acetate media. Let's see the principle that how this test will be working. Now this test, as we talked, that in this test we have mostly acetate. So just like citrate, in this test we have what? We have the acetate, and it is mostly used by the by the bacteria, due to which we can easily differentiate the member of Enterobacteriaceae family. Next point says that moreover, there are different types of the bacteria that are using organic acid in the presence of organic nitrogen. Third point says that the acetate media, as we talked, it contains inorganic ammonium salt, okay, and it is acting as a nitrogen, source of nitrogen, along with acetate that is acting as a source of carbon. So ammonium salt is acting as a nitrogen, while acetate is acting as a source of carbon. Next point says that now remember, when the bacteria or any microorganism in the media that is growing, so what will happen? It will utilize the acetate, and when the acetate is utilized, so then the test will be positive. And what will happen? The utilization or the metabolism of this acetate will break down the ammonia. So when the ammonium is break down, so then when the ammonium salt is break down, so then it will be converted into ammonia. Okay, it is converted into ammonia. Now what happened after that? After that, the ammonia will increase the pH of the media. Okay, so when the media pH is increased, so what happened? We have an indicator which we called it bromothymol blue indicator. This will be changed into green to blue. Okay, so that's why the whole media is working, the whole test is working. This is the principle. Next we have the material that is required to perform that test. So we need PPEs, that is the personal protective equipment. So in this case we need goggles, gloves, and we need shoe cover, we need overall. All these things are mentioned in the personal protective equipment. We need a spirit lamp burner, we need fresh bacterial culture, okay, test tubes, wire loops, or inoculated needle, sodium acetate culture media. So all these things we need. There are some other material but I forget, but the material which I mentioned here, these are the important. Let's see the procedure, that how to perform that test. First of all, what will you do? You have to prepare and sterilize the media. I hope you know the sterilization, each and everything. It's really simple, but what will you do? First of all, you will have to weight the media powder and will mix it into the distilled water, and then you will have to clave it at 121 degrees Celsius for 30 minutes. Then after that you will pour it into the sterilized test tube. Remember, that point is really important, sterilized test tube. And what will you do? You make butt and slant. Now how the butt and slant will be made? You have to put, this is, let's suppose, the test tube, so you have to put the test tube just like in this position. So what will happen? Let's suppose this is the test tube which is present in this position, and this is the media. So after, when you put the test tube in this position, so what we get? We get the slant position. So this is the slant position, as you can see. So now this is the slant position, this is the butt position. So you have to put the media like in this position. Okay, now after that, what will you do? You will inoculate a fresh bacterial culture, and then you will incubate the tube by losing the cap. That's really important, because this process is mostly performed in aerobic condition. If there is anaerobic bacteria, so it will be grow in the bud portion. That's why we also providing the anaerobic condition in this test as well. Now next point says that if you want to test the Enterobacteriaceae, so you have to incubate it for 5 days up to 7 days, and it is important for the non-fermenting gram-negative rods. That's why the time is a little bit longer. The next point says that after every 4 days you have to check the tubes for the positivity. Sometimes the result will be we get the result after 2 days, 3 days, 4 days. Okay, so within 7 days we will get the result. After the 7 days, if still we didn't get any result, so we will discard it. So that's how we can perform this test. Let's see the result. You can see this is the positive result, this is the negative result. A positive result represented by the growth and change from green. Okay, so this is actually the green media. When it is converted into blue, so it means the result is positive, and it is the E. coli that convert it into green to blue. Negative result is that if the media is the same, mean if it is green and after the inoculation, after incubation, we get the same media, so it means that the result is negative and the bacteria will be Shigella. Now let's see the limitation of this media or this. You observe the growth on the slant position, and it will indicate the positive result. So remember, this type of the mistakes are mostly occur, and it is due to the heavy inoculum. What does it mean? It means that when you want the correct result or the best result, you have to perform the test with less inoculum. That's really important. The next point is says that the test should be repeated if you have any doubt. Next point says that the slant should not be steep. Why? Because if you require the aerobic environment, that's why the slant should not be stepped. Next point says that you should have to take the fresh culture from the agar plate, not from the broth. The next point says that a light inoculum should be taken. Why? Because it will prevent the false result, or prevent the carryover of the substance from the previous media. That's why you have to use light inoculum. Next point says that there are many E. coli strains, next point says that there are some strains of the E. coli that can use the acetate slowly, and due to this way, what we get the negative result, false negative result. So you have to be very careful. Although the result will negative, but it is actually positive. Why? Because the E. coli is present, but it is actually utilizing the acetate slowly. So there are some strain we have. So that's all about the acetate utilization test. I hope this video is helpful for you. Thank you so much for watching.